in Alzheimer's disease. So I'm sure everyone attending knows quite a lot about Alzheimer's disease, likely more than myself. So I will not waste time repeating what you already know, but try to focus on what you likely do not know, save for a quick introduction on amyloid beta and tau. So the two histopathological hallmarks of Alzheimer's disease are extracellular amyloid plaques and intracellular tau fibrils composed of amyloid beta and hyperphosphorylated tau protein, respectively. Up until now, the amyloid hypothesis has been the prevailing model in Alzheimer's research. It was believed that accumulation of amyloid in the extracellular space of neural tissue causes neural loss and neurodegeneration. Consequently, most drugs in clinical trials up until now targeted amyloid, all of them failures. The long line of failed anti-amyloid drugs has called into question the amyloid hypothesis and has prompted the research community to turn to other potential targets, one such target being precisely tau. The mechanism of tau accumulation and fibril formation inside neurons is not known. It is currently believed that tau is condensed by cytosolic polyanions, namely heparin and RNA. However, once I became familiar with this hypothesis, I immediately noticed three major flaws. First, heparin is overwhelmingly an extracellular substance. Very little, if any of it, is present in the cytosol to interact with tau. Second, most RNAs have very short half-lives, on the order of minutes for most messenger RNAs, with what longer lived RNA being inside cells being sequestered away from tau in ribosomes, stress granulae, and nucleoli. Thus, it seemed to me that it's highly unlikely that tau condensation of with the tau, that RNA or heparin-mediated tau condensation occur in vivo to any significant degree. Lastly, this hypothesis does not take into account the presence inside cells of polyamines, essential polycationic substances that have multiple physiologic functions, including condensing DNA inside the nucleus. To show that hyperphosphorylated tau is condensed by polyamines, in the absence of experimental data, we performed molecular dynamic simulations on 8 hyperphosphorylated tau chains with spermine, 8 unmodified chains with spermine, as well as the same systems without spermine, so 4 systems in total. I'm sure most people attending today are doctors, biologists, or neurologists, so I will spare you the gory simulation details and try to focus on the major takeaways. So we first analyzed the radial distribution function of spermine heavy atoms around protein heavy atoms. To make it short and sweet for you, values below 1 indicate that spermine is less likely to be found around tau at the given distance as opposed to bulk solvent. Values above 1 indicate the opposite. So briefly, we see that spermine is less likely to be found around unmodified tau and more likely to be found around hyperphosphorylated tau. Sodium behaves similarly to spermine, positioning itself around phosphorylated tau and away from unmodified tau. Somewhat surprisingly, chlorine ions appear to be non-selective, positioning themselves away from both forms of the protein, despite the abundance of positive residues and the high negative charge of unmodified tau. Comparison to the spermine-free systems allows us to assess the behavior of the ions, the effect of spermine on the behavior of the other system components. Briefly, we see that ions behave quite similarly in the spermine-free systems compared to the spermine-containing systems. The major notable difference being the higher peaks of the RDF values for sodium. This indicates that spermine competes with sodium for protein binding sites. We next observed the effect of phosphorylation on the propensity of tau to form intermolecular bridging interactions. An intermolecular bridging interaction is where an ion water molecule or spermine molecule is simultaneously hydrogen bonded to different tau chains. So briefly, the top row indicates the number of the bridging interactions per frame. The bottom row shows the duration of the bridging interactions on a semi-log graph, so between 0.1 and 1 nanoseconds, 1 to 10, 10 to 100, etc. Briefly, we see that phosphorylation greatly enhances the number of bridging interactions, specifically sodium and spermine-mediated bridging interactions, as well as their lifetime. If we now compare the spermine-containing systems to the spermine-free analogs, we see that the behavior of the unmodified system is essentially unperturbed, whereas the number of sodium-mediated bridging interactions increases nearly 8-fold. Water-mediated bridging interactions are only slightly affected. This confirms that spermine competes primarily with sodium rather than chlorine or water for protein binding sites. Okay, this slide is pretty straightforward. From the simulations, we directly computed the diffusion coefficient of spermine around phosphorylated and unmodified tau, and we see that spermine has a nearly 100-fold lesser mobility around phosphorylated tau than unmodified tau. So at this point, a clear picture begins to emerge. On the one hand, we see that spermine, shown here in stick representation, and the ions, shown here as spheres, prefer to position themselves away from unmodified tau. And this is shown here on the left panel, where we see a very zoomed out view of the system with the polyamine and the ions away from the protein. Conversely, in the hyperphosphorylated system, spermine and sodium position themselves around phosphates and carboxylates, shown on the right as sticks. We also performed MMGDSA calculations, and we performed per-residue decomposition analysis to get a more quantitative assessment of the affinity between spermine and unmodified and phosphorylated tau. On the left, we see that for the unmodified system, the energy of interaction plateaus around 0 near the end of the simulation, which indicates that it is energetically more favorable for spermine to interact with solvent rather than with tau. Conversely, for the hyperphosphorylated system, the energy of interaction is large in magnitude and negative in sign, indicating that this is a highly favorable interaction. This is the per-residue decomposition analysis that I mentioned. So, blue indicates a favorable contribution to binding, red indicates an unfavorable contribution to binding, white indicates no contribution to binding. So we see that the major contributors to binding are phosphates and carboxylates. The two systems exhibit drastically different behavior despite the same starting conformations. So we present evidence that spermine has high affinity for hyperphosphorylated tau. We present computational evidence. This is important because a higher affinity for each other than the remaining system components could drive tau-mediated, spermine-mediated phase separation. We see it is notable that during our MD simulations, beta strands started to form. Although our initial structures contained no beta strands, we observed several forming early on into the simulations and persisting throughout. This indicates that what you're seeing here could be what the inside of a tau-rich droplet looks like. Densely packed phospho-tau chains with spermine and sodium ions mediating the tight packing. It could be that the increased concentration of beta-prone stretches of sequence in such tau droplets serves as a primary nucleation center for the formation and growth of beta-rich fibrils, transforming the innocuous soluble phase into cytotoxic beta-rich fibrils. I'm not going to discuss the concentrations in here. The question of concentrations, if anybody is interested, we can get into it. So this has implications for Alzheimer's disease target discovery that I'll get to in a bit. It seems, however, unlikely that tau condensation is mediated by RNA and heparin for the following reasons. First, tau tangle formation is strongly associated with the appearance of hyperphosphorylated tau, and heparin and RNA condensation of hyperphosphorylated tau are unlikely, first and foremost because of electrostatic considerations. Then, if you consider unmodified tau, there is the scarcity problem inside cells of heparin and RNA. Rather than native tau condensed by polyanions, our work points to phosphorylation mediated charge inversion, with hyperphosphorylated tau being condensed by polycations. And now, on to the target discovery implications. If our proposed mechanism is correct, one should first have a look at kinases and compare kinase sequences between Alzheimer's disease patients and healthy controls. I suspect that one or more mutated or hyperactive kinases, or one or more mutated or hypoactive phosphatases could be, if not the root cause, then certainly involved in the root cause. Now, of course, you should probably say to yourself at this point, there's over 500 kinases in the genome. That seems like a pretty daunting task. Well, obviously, one could narrow down the search by first focusing on neuronal enzymes. I doubt there's more than a single digit or low double digit number of kinases expressed in neurons. Moreover, it is well known that Down syndrome, trisomy 21, and Alzheimer's share many features. Amyloid plaques, neurofibrillary tangles composed of hyperphosphorylated tau, which leads me to believe that it would probably be a good idea to start searching for kinases in chromosome 21. Recently, I became aware of a kinase, DYRK1A, located in chromosome, where it was, I came across a paper where it was pharmacologically downregulated, and a lot of the tau related symptoms, such as tangles, were ameliorated. That was, I think, cellular or mouse models, but still an interesting find. There's also the case, it's also the case that amyloid precursor protein is located in chromosome 21. Okay, at this point, it would probably be good to wrap up and thank the Drug Design and Bioinformatics group at the Medical University of Sofia, where I did all this work. To thank you for your attention, and I will now gladly take your questions. Any question?
You're looking at me, but you already
you
already answered my question. Thank you. It's about.
Question?
Yeah.
Question?
A few questions. Not being controversial with your presentation, that's very interesting because it's really challenging to understand how tau is aggregating and the very beginning of aggregation of tau. And as you said, kinase and DYRK1A is one of the potential targets. GSK3 beta has been considered as also as a target, and also phosphatase has been considered as target. But did you ever consider that you may have several populations of tau inside the cell, in terms that there are some hyperphosphorylated tau, but there are also some unphosphorylated tau that can be mixed together?
Yes.
And this was really the hypothesis by Iqbal, who showed that if you mix those two kind of population, you don't need to add HS or RNA or whatsoever to make tau to aggregate.
So natively, there's a balance between phosphorylation and dephosphorylation, and different kinases phosphorylate tau at different sites with different efficiencies. So in vivo, you have a mixture of different isoforms of tau and differently phosphorylated forms of tau. However, you constitutively have polyamines inside cells. So polyamine, they're both in the cytosol and in the nucleus, and they never go away. So in the healthy state, there is a balance between phosphorylation and dephosphorylation, so the tau is phosphorylated to a level of 2, 3 to 4 moles of phosphate per protein. Only when phosphorylation turns to hyperphosphorylation, when it comes to dominate, and you get 10 moles of phosphate or more per mole of tau, do problems begin. So I suspect the first thing I suspect and would like to check is kinases and phosphatases. It could be that it's not even a mutated kinase, it could be just a hyperexpressed kinase. Say it's something in chromosome 21, you have 3 chromosomes and you have too much tau phosphorylation going on, and we know what happens then. If that fails, the next thing to check would be factors that directly regulate kinases. So it's maybe not even a mutated or not even a hyperexpressed kinase, it's a mutated regulatory factor that is driving up the kinase activity, and you end up with too much phosphorylation. Then if it's not a level 1 factor, it could be a level 2 factor and so on, but that quickly leads to an explosion of search space. So I would be happy to start with by looking at neuronal kinases. And in the case of glial tauopathies, obviously you would want to look at glial kinases. And I'd be interested to see what turns up.
Just to push a little bit the reflection. You said that polyanions are found in nucleus.
Polyamines are in the nucleus. And polyamines, they condense DNA. Otherwise you can pack it.
And tau binds polyamines in nucleus because tau is also regulating double-strand break DNA and also contributes to stability of the genome. And this has been shown. Yes, tau.
I was not aware.
And it never aggregates inside the nucleus. Aggregates of tau have never been observed in the nucleus.
I'm not surprised. So you will need hyperphosphorylated tau, and phosphorylation occurs in the cytosol. So that fits in very well with my model.
Okay, thank you. Other questions?
I think I have a question. By the way, you had answered my question before, which is what I think. Something else just popped up. Tau proteins of this kind are associated with a whole host of neurological problems. Pick's disease, supranuclear palsy, and a long list. One of the reasons that I don't do clinical work is because I can't get my head around it. So if tau proteins are associated with chromosome 21 and Pick's and supranuclear palsy and so on and so forth, maybe there's something underneath, one common thing underneath all of it. That's one of the problems that I also have with a neurological differential diagnosis, which is, so what's the difference between Parkinson's and MS and degenerative conditions apart from 17 and other conditions without? The answer is, I don't think there is. then I really think that neurodegenerative conditions are neurodegenerative conditions, and you may be underneath the whole host of all of these things. And I think what we need to do is, what clinicians need to do, is to differentiate, because maybe they're targeting drugs from one area of the brain and not to another area of the brain. So I guess my point is, maybe it's not about Alzheimer's, maybe it's not about PICs, maybe it's about degenerative conditions of the brain associated with dementia. And it's got something more to do with dementia and with progressive destruction of brain tissue than it has anything to do with Alzheimer's. Maybe that's where what we're missing, maybe?
Maybe, but this we can maybe further discuss after you talk.
Okay.
All right, am I going overtime? I'd love to discuss, but am I going overtime here?
Are there other questions? So we will continue discussion at the coffee break.
Oh, all right, thank you very much.